All right, so get your SnapGene up and running. So on the website, there is DNA, ProK1, EGFP, but we can start out by learning how to import DNA. So first thing we'll do is, let me show you a website, addgene.org. So AddGene is a non-profit plasmid repository. They hold a ton of plasmids, which they send to academic researchers and non-for-profits. Um, it's very difficult to get them if you're a company or other things. The way they send them is they use a material transfer agreement, and the material transfer agreement usually prevents them from being sent to companies. But what you can do is, if you have a local biohacker space or know people at a biohacker space, you can contact them and order plasmids through them. You might even be able to contact AddGene and talk to them about ordering through them. But it's pretty inexpensive for the plasmids. So generally, plasmids are $60, I think, or $65, um, which is it's not extremely inexpensive, but it's pretty reasonable. And they send it to you as a bacterial stab, like we are doing in this class. We're sending the DNA as a bacterial stab. So you're going to learn how to purify it and grow it up and use it and all that good stuff. So SnapGene and AddGene are connected. So if we go to SnapGene and we can go import sequences, one of the best places to import sequences is from AddGene, because usually if there's a plasmid, you got it from an academic or somebody who got it from an academic. The plasmid is probably also on AddGene. So the first plasmid we're going to look to is this plasmid we got from George Church, and it's called ProK1. ProK1 EGFP. So the ProK1 is in reference to the promoter that's used, and EGFP, I think E is enhanced GFP. But what you can do is you can just search for this in the import and just click import. It'll download the file for you and open it up. So this is one of the plasmids we're going to be working with in class. So either download it from the Google Classroom website or download it here and open it up. And we can take a look at the plasmid. Right, so I'll give you a second to do that. So once you open it up, there are two main views we're going to use. One is the map, the other is the sequence. The sequence is, as you imagine, it's the exact DNA sequence that's used, so a bunch of A, T, C's, and G's. And the map is just like a higher level view, so it has these elements, genes, and other DNA elements that are identified by the software that let us know what's going on in this plasmid. And we will go over those. So, everybody good? Everybody, you got your plasmid downloaded and SnapGene downloaded and things ready to go? Maybe we should try doing this on Zoom. if the classes are going to be reasonable size, because then we could get you know, real-time feedback. I feel like YouTube, the feedback is delayed a bit. The stream is delayed and the feedback is delayed, so it's like, uh, all right. So I guess I will assume that we can go forward. For the sake of the stream, I will. All right. So there are, let's say, three major parts to a plasmid. The first is the antibiotic resistance gene. Now, there are some plasmids with antibiotic resistance gene, but in the majority of cases, every plasmid has an antibiotic resistance gene. This is because all plasmids generally are replicated in bacteria, because bacteria are easy to grow, and they love making plasmids, and they're easy to purify plasmids from. So plasmids will generally have an antibiotic resistance gene, almost 99.9% of them. Now, in some cases, if you have, if you have a plasmid that works with mammals or humans, they might have multiple antibiotic resistance genes. Um, so, like for instance, this one has a neomycin resistance gene for mammalian cells and an ampicillin resistance gene for bacterial cells. So, the neomycin resistance gene allows it to be selected in mammalian cells or human cells because neomycin will, kill or inhibit human cells from growing, and ampicillin will inhibit bacterial cells from growing. Plasmids in plants also, they have their own specific genes that are used for antibiotic resistance or whatever. So like depending on the organism, they might have multiples, but usually always will have the bacteria antibiotic resistance gene. So this is pretty much an essential part of the plasmid. Now, that's because like bacteria and other organisms, they generally don't like plasmid DNA. So if you give them plasmid DNA, they're just like, what the fuck is this? I don't want this, and they'll just get rid of it. They won't replicate it, they won't use it. So you basically have to threaten their life. Um, you have to threaten their life, like, I'm going to kill you unless you use this plasmid, right? And then they will use it. Then they will use the plasmid. Um, but otherwise, that's pretty skeptical. Now, there are some cases, like if you take the bacteria that you have and you streak it out on LB agar without antibiotic, usually sometimes they will keep the plasmid. After a couple times, couple generations that you streak out, they'll probably lose it, but sometimes they will keep the plasmid after you get it into them for a little bit. But they generally don't like to. Now, there are a couple different antibiotics. I think in this class we're using ampicillin and kanamycin, but there are many other ones. There's a whole repertoire. One place that sells reasonably priced antibiotics is called Gold Bio. They're probably going to find the best prices. And Gold Bio, I think, usually ships to anybody. Or, just make up a company name and have them ship to it. But you can get most anything uh, from Gold Bio. $23 for 5 grams, not bad. $34 for 10 grams. Another trick is that sometimes on eBay they sell antibiotics for fish and other animals and stuff like that. So sometimes you can find uh, antibiotics on eBay. Not always, but sometimes. Like here, look, you can find fish ampicillin, um, or pigeon. This one says it's for pigeon. Uh, it's okay. It's legal, I guess, to sell these for animals, but not for humans. So yeah, they sell them for fish. So if you're ever in need of certain antibiotics that you're having trouble find, always check on eBay. Sorry about that. Yeah, definitely. Shipping to a UPS mailbox or a PO box always helps out a bit. It gives you kind of like a business address. So that's you know, a biohacker trick is that if companies won't send to your home address, just open up a PO box or a UPS mailbox. They're usually pretty inexpensive, and then you could get places to ship to you. All right. So antibiotics, that's the first one. Now the other one is the origin of replication. And you can see we got a little bit more information here. It says it's a high copy number, uh, COLE1 PUC origin of replication. What does that mean? Who cares? Whatever. So, origin of replication basically tells the organism that they need to replicate this piece of DNA, right? So, basically, what a plasmid is, it's this piece of DNA that's trying to trick the organism into thinking that this piece of DNA is its own DNA. You're basically just like hacking the, I can't believe I'm going to say this, but you're basically hacking the organism's genetic operating system. Um, that sounds so terrible and cheesy. And the origin of replication tells the organism to replicate. Now, there's different so yeast have different origins, mammalian cells have different origins, all different origins. Bacteria even have multiple different origins. So what we can do is if we look up origin of replication. Now, origin of replication, um, AddGene is usually a good resource. Scroll down here. Here we go. So, origin of replication, if it's a high copy number, it literally means number of plasmids inside the bacterial cell. Sounds weird, I know, but inside each bacterial cell, there's not just one plasmid, there's usually many plasmids. So, generally, you just want the highest copy number you can because you're maybe using bacteria to express a protein or to purify DNA from it. So generally you go for a PUC or a COLE1. I think people call it COLE1, but I don't like that. COLE1 promoter, and that's what we have here. Now, this will give us the most amount of DNA, which generally gives us the highest amount of protein expression and the highest amount of DNA that we can purify. Now, why would you want a lower copy number piece of DNA? Now, sometimes you're working with proteins that either A, might be toxic to the cells, or B, when there's large quantities of these proteins, they tend to aggregate and they don't function properly. So you might want to produce a lot less of the proteins on your plasmid. And in that case, you'd go for a lower copy number origin. Generally, these sequences can just be found online by, searching the internet. So just search the internet if you're looking for these other sequences. But also, one thing you can do is you can just go to AddGene, what I like to do, and you could just say, let's say PBR322. We want a plasmid with the PBR322 origin. And you could just pull up a plasmid with PBR322. Wow, that one looks old and doesn't look like it. Or maybe it does have a sequence. Can we filter by sequence? Let's just filter by popularity. So we can pull this up. Let's see the sequence. Okay, so we can even look at the sequence on AddGene, which is pretty cool. And we can find the PBR 322 origin, right? This one actually looks like it might be a high copy number origin. But you get my idea. You can search AddGene and find general sequences that would be in plasmids from AddGene by just searching for the name of the element. So the origin of replication, now I think Bill, William, Bill, you go by Bill, right? Bill asked a question in the classroom. Getting a message, it says, could not be important because only partial sequence data are available. Joe. So Joe, if you're running into trouble getting that sequence, what you can do is you can go to our classroom website, and the sequence was put up on the classroom website, and you could download it from the classroom to Google Classroom, and then you can just open it in SnapGene from the Google Classroom. So try to do that. Try to download it from Google Classroom. So now Bill was asking, can I put different plasmids or more than one plasmid or inside an animal or bacteria or something like that? And the answer is yes, you can. So there used to be this idea that you couldn't have two plasmids with the same origin of replication because uh, like the plasmids wouldn't all get replicated if they had the same origin of replication. AddGene even has this little incompatibility group table here saying that like all the ones with A can't be used with A. They can only be used with B or C, etc. Well, in the past, I don't know, 5 years or so, 10 years, people actually started testing this out, and they're like, that doesn't really make any sense. And they started just doing experiments with plasmids that have the same origin or replication. They found it works. So you can have multiple plasmids in an organism, and you can have multiple plasmids with the same origin of replication. Uh, it's best if you have two plasmids with a similar origin of replication that you have different antibiotics that you select for, right? That'll just make it more likely that the organism keeps two plasmids. I don't know what the record is, but it's pretty easy. It's commonly done to get two and even three plasmids into an organism. Beyond that, I don't know if anybody's really tried or how much people have tried, but yeah, it doesn't seem. Yeah, Jeff. High copy number means after each generation, a bacteria will have that many copies. It's yeah, generally. I mean, I don't know if anybody's looked at the exact timing of it, but when the bacteria replicates, it should replicate approximately that many copies of the plasmid and produce copies over its lifetime. But, uh, all right. So that's what the origin of replication are. Now you have to understand in plasmids, there's usually a lot of what I call, just like void, black space, black hole. So a lot of plasmids were made from old plasmids that were made from actual plasmids that like so a plasmid isn't a thing created by humans. It actually exists in bacteria by itself, and people kind of just took elements and left them in. So plasmids aren't like a very tight piece of code. Usually they have a lot of elements that either are left over from something or are just around as junk. Now, it's not always the case, right? So you can't just like take a plasmid and say, oh, I want to remove all this stuff here. I want to remove ROP and bomb. I want to remove the F1 origin, though you could probably easily remove the F1 origin and nothing will happen. I want to remove this space in between the AMP-R and the origin. But generally, you can't and you don't want to. If you're designing a plasmid from scratch, you can mess around with these things, but these spaces sometimes tend to have either spacer regions that the organism needs to have space between these genes for replication, for transcription or translation or other things, or it's just space that we don't know what it does, right? But it does something. And I don't think anybody's really systematically gone through plasmids and, you know, tried to make a most minimal plasmid possible. But there are definitely plasmids that are much, much smaller than this, right? So this plasmid is not the most minimal plasmid possible, but it's just somebody made it. It's easy to work with. It's hard to delete all that other DNA that's in there. So we're just going to leave it be. Now, the last, generally the third part component of the plasmid that is used is the gene of interest, or RNA of interest, or the thing that you're actually trying to use or express in the plasmid. So these other things are just kind of like what makes the plasmid work, right? They're just, the base code that makes the plasmid work. And then on top of this base code, you can put in the things that you want to work. And in this plasmid's case, that's the green fluorescent protein, GFP, which makes the bacteria fluoresce green. Yeah. All right. So we doing good? All right, we're doing good. I don't know what those three black. Oh, those are holes. They look like black footballs, Craig. I couldn't figure out what that was. All right. So now, after we looked at this, what we're going to do is we're going to start looking at the sequence. We're going to zoom in, so let's go to the sequence. And let's go to this green part of the sequence where the GFP is. Now, I think this plasmid is actually missing the promoter. Let's see what happens when I detect common features. Nothing. Oh, no. It's all the same. File. Let's see. Open recent files. ProK1. Let me see if I have a copy. So what this plasmid seems to be missing, we might just be able to find it on AddGene. So let's look on AddGene, ProK1. All right. ProK16, ProK1. So what I'm looking for is the promoter element. So sequences need promoters. When you're trying to express a protein or a sequence, you need a promoter. A promoter tells it tells the RNA polymerase where to start transcribing the DNA. So let's see. Analyze the sequence. Maybe it's in here. Features. Doesn't have it in here either. Where's this? Alright, let me search on myself. Open. Google Drive, Plasmid. Let's see. ProK1. So I just downloaded that ProK1. Do I have another? Well, this one's from 2000 and what is this one? It's different? No, maybe it just doesn't have the promoter, which we're gonna have to find. My bad. I'll probably have to find that at another time. Wait, hold on. Let me see. Let's see if we could actually just, this is a good experience. So what I'm looking for is the promoter to this GFP sequence. That's actually what ProK1 stands for. So what we can do, let's go to the original paper and see if we could find this promoter. So I just click through on AddGene. If you click on the publication, so it gives you the publication that the plasmid is from, which is nice. We go on here, and now we can look and see where the promoter, 5 promoter libraries. All right, here's the promoter sequences. Let's download this, open it up. All right. So ProK1, TTTACT. That's where it starts, I assume. TTTACT. Let's see. Now what we can do is search FTTTATC. Find. Whoa, that's not it. Nope, nope, nope. Did I do that right? TTTACT. ACT? Not ATC. All right, there we go. All right. So this, what I imagine in this ProK1 sequence, TAATAT. I think that is the Tata box. TAA. Let's go. TAATAT. Right there. So this is the sequence from ProK1. I think this is the whole promoter, but I'm not 100% certain. We'll have to look into it. But so each sequence, what they do is they have a promoter region before it. So if we look at, say, the ampicillinate one doesn't actually have a promoter on this plasmid. It does, but it's just not being identified. Let me pull up another plasmid and show you. I think one I was just looking at. So generally you have these promoters, right? So even the antibiotic resistance gene has a promoter in order to tell the organism that it needs to create this sequence, right? So AmpR has a promoter. I'm sure we could actually maybe even search for this in our plasmid. Maybe. Nope, it's not being found. It's probably some version of this promoter right here that makes this sequence. Anyway, it's not identifying it for some reason in the software. But just, trust me in that when I'm saying that these genes have promoters that are before them. Now, the other thing that is important is the ribosome binding site, RBS. Now, the ribosome binding site, what this does is it tells the ribosome that it should bind to this RNA and start making a protein, right? So if you remember your molecular biology, ribosomes bind to the mRNA and start to make proteins. The plasmid from the classroom is missing a few components. Okay, so let's see. Let me see. So what you can do is go to features, go to detect common features, and then it should pop up something that should detect the common features. And you should be able to add those to you should be able to add those to the thing. Let me check on the classroom. Classwork. It's also completely possible that I uploaded the wrong file on accident, but it doesn't look like it. It looks like it's the same name. Let's open it up. ProK1, EGFP. It looks the same. So the difference is it has the AMP-R name. Instead of AMP-R, it says BLA, which is beta-lactamase resistance, which is ampicillin. So it's the same thing. And the colors are different. But otherwise, it looks the same. What are you all missing? Is there something you're missing? I think it might just be the colors and things like that, maybe. So let's see, in my SnapGene map. AMP-R, origin, GFP, yeah, and just instead of saying those, maybe it says a yeah, Jeff. So after the promoter, yeah, no, so transcription doesn't begin at the first start codon. Translation begins at the first start codon. Transcription begins generally after the promoter and ends at the terminator. So generally what you'll have is, if we look at the sequence, I don't know why this isn't fitting. And so if we look at the sequence, so we have the promoter before this in the ribosome binding site, and then after the DNA sequence, what we have is we have a terminator, right? And the terminator tells it when to end transcription. You could leave out a terminator technically, but what happens then is that the RNA polymerase will keep transcribing the DNA and eventually just fall off. It'll just give up and fall off or something. So what happens is it becomes a lot less efficient. It'll make a lot less of the gene that you want made. It's a cool trick that you can use if you want to maybe have two genes made at the same time or things like that. That can work. If you put one gene after another with a stop codon and a start codon. But generally, you want both a promoter and a terminator, and they will tell the RNA polymerase when transcription. Yeah, exactly, Jeff. So the translator is from start to stop codon. If the match percentage is really high, does that mean it is already in the sequence? I don't know what that means. Forward your nuance, sorry. But yeah, okay. So we're all Getting there on the same page, right? We're all getting back. So we have our gene. So generally plasmids already have all this stuff, and all you want to do in your plasmid is insert your gene of interest, right? So the other plasmid we're going to be working with, if we go to import, let's go to AddGene plasmids and try this. We're going to be working with this plasmid pDawn. So let's get pDawn, import it, and we could look at that also. Now, immediately you can look at it, and from what we know, right? So we know this one is kanamycin resistant. We can look at the antibiotic resistance gene, right? This one's kanamycin resistant. The other one's ampicillin resistant. And the origin, it's a high copy number origin, right? So we'll make a lot of this. And we can look. Now this plasmid, the one we sent you actually has a gene in it. It has a red fluorescent protein. But in this case, this is just the plasmid without the red fluorescent protein. So the gene would be between the RBS and the 6X his. But that's where the gene would generally be. But you can put it anywhere, again, between the terminator and the RBS. Now this plasmid is a little interesting and crazy, and we'll get into it more later in the class because it's a really complicated plasmid. There's a lot of different things going on. So don't get too confused by it. But it's just an example of another plasmid and something to look at. So we'll go back to the ProK1 GFP. Now, you're probably wondering, well, all right, we got a plasmid, we purified our plasmid, and we want to put a new gene in this plasmid. How do we do it? Now, the way we're going to do it in this class is a method called restriction enzyme cloning, right? Now, you may have noticed that if you look at the map and you see all these weird letters and numerals around the whole thing, now these are restriction enzymes. Restriction endonucleases, or endonucleases, they're also called. Now, what these are enzymes you can buy from companies, and they cut DNA. They cut DNA at specific places, right? So, for instance, if we wanted to cut out the GFP gene and put in another gene, we would try to select a restriction enzyme that's somewhere before the GFP gene. So say XBA1 and a restriction enzyme that's after the GFP gene. XHO1 or BLP1 or something like that. So once we have selected the restriction enzymes, what we can do is we can mix the plasmid with these restriction enzymes, and what they'll do is they'll cut the plasmid in these two places. They'll cut at the XBA1, and they will cut at the XHO1, and the GFP gene will be completely removed from the plasmid, right? Cut on both sides of it, and it comes out. It's a pretty cool trick. It's, you can imagine having to find all these enzymes and companies purifying and making all these enzymes just to do that seems pretty arduous. There are easier methods to do, well, I don't know if I would necessarily say easier, but there are less complicated methods to do cloning, but generally restriction enzyme cloning is the standard. So now, where do you get restriction enzymes? So there are companies. A big one is NEB, New England Biolabs, and they basically have every restriction enzyme you want. Now NEB, sometimes people have trouble ordering from NEB. to ship to their home. It's a lot easier to get it shipped to a PO box or another box. But if you search the internet for restriction enzymes, you can find a lot of companies that sell restriction enzymes, and that will probably sell to you. You generally can find one that will sell to you. Now, one thing that you have to be clear with is this sequence doesn't necessarily display every restriction enzyme. What it displays is some of the more commonly used restriction enzymes. But if you wanted to look at all restriction enzymes, what you can do is you can go up here, you can go to choose enzyme or even use enzyme set, and you could select the restriction enzymes that you want. Now, in most cases, you want restriction enzymes that are unique cutters, right? If a restriction enzyme cuts in two places, right? So if you have dual cutting enzymes, what happens is it'll cut in two different parts of the plasmid, and that's not what you want. You want restriction enzymes that cut in single parts of the plasmid. What we can do is we can choose all commercial restriction enzymes, and you can see, holy cow, that's a lot. But this gives you a lot of things you can do. So here's all restriction enzymes. Now what we see here, the ones that are in bold, that indicates that they're single cutters. So there's only one site in the whole plasmid. The ones that aren't in bold, that means that there's more than one site in the plasmid. So generally when you're doing this, you want to choose a restriction enzyme that only has one site, or the two sites are within the same region. So, for instance, on PDAWN, if we look enzymes, let's choose enzymes. Let's type in XBA1, add XBA1 here, click OK. If we were trying to use XBA1 in PDAWN, oh, where's this? There's a restriction enzyme before. There's two restriction enzymes before the gene. So if you used it, it would cut out this lac repressor gene. Now, we'll talk later about whether you want the lac repressor or you don't want the lac repressor in there. But theoretically, you could totally use XBA1 and just be fine because it's not cutting out anything you need necessarily, and it's cutting out something before the gene. But these are, rare cases. In general, what you want to do with enzymes is you want to focus on unique cutters, right? You can look at New England Biolab's Thermo Fisher, but we'll just go for unique cutters. And unique cutters in here. So the goal, one of the goals of this class is going to be to take the GFP gene out of this plasmid, put a new gene in, and put the GFP gene in another plasmid. So we're going to try to swap around genes in plasmid DNAs so we can learn how to do DNA cloning. Yeah, no, it is. It is if you need to make genes, if you need to make genes or find genes, uh, if you want to get like a GFP oligo or something like that, people do use this way where they'll just restriction digest out parts of plasmids. It's common in CRISPR. So sometimes in CRISPR, when you want or need template DNA, sometimes they'll just grow up a lot of plasmid and restriction digest it, and then use that as a template in your CRISPR reaction or transformation. But yeah, it is. Generally, you want to use PCR or something, but sometimes it's a lot easier just to grow up a lot of DNA and bacteria and purify it. That's something we'll go over in this class. We'll learn how to grow up bacteria and purify it using mini preps and mini prep columns and things like that. All right. So let's see. Is there anything else we need to talk about in GeneWiz today? Anything else we're going to use in this class? I think those things are mostly it. You know, enzymes and sequences, um, promoters and terminators and things like that. The one, I guess, final thing, one thing we're going to be doing is DNA sequencing. Now, company that we're going to be using in the US is called uh, GeneWiz. So genewiz.com. Yes. So they do, let's see, Sanger sequencing of purified templates. So they have a list of the primers that they have. So generally, every plasmid has these sequences built into them because these sequences make it easy to sequence the DNA. So one thing you're going to do is after you put in your gene of interest, you're going to want to sequence the plasmid to make sure you have the correct sequence inside the DNA. All right? So what you can do is you can search for these. So if you're sending your DNA off for sequencing, like we're going to do, you can say, oh, it should be the T7 terminator. But you can look for these. Generally, the ones in bacteria are M13 and T7. Sometimes you'll have these other ones, but generally it's M13 and T7 in bacteria. And see, so what we have a match here is for the T7 terminator primer site. So what happens in DNA sequencing, we'll go over DNA sequencing. The DNA will sequence starting from this and go backwards and give you the sequence of all this stuff that's in there. It'll let you know, did my sequence make it into the plasmid? Is it correct? Does it have any mutations or other things like that? Because, you know, replication errors do happen in organisms and bacteria. So you are going to get replication errors. You are going to get sometimes the sequence that you don't want. Sometimes you're going to get weird artifacts, which maybe we'll see in this class, which will be fun. Sometimes just like weird stuff happens with DNA that you can't explain because something doesn't get digested all the way or something else happens. But, uh, you know, so you always, always, always want to sequence DNA. I think the first thing we're going to do is purify the DNA and sequence it in the class, if I remember correctly. Please forgive me because I'm old and tired. But whenever you get plasmid DNA from anybody, doesn't matter if it's your best friend, send it off for DNA sequencing. You just it costs $6 or something like that, and it lets you know that your DNA is good because you don't want to go through weeks worth of experiments only to find out that the reason your DNA is not working is because your buddy accidentally gave you the wrong DNA or it was contaminated by something or something like that. So always, always, always sequence the DNA you get before you use it. Doesn't matter if George Church gives it to you. Sequence it. Sequence it. Always sequence it. All right. So do we have any questions about the plasmid that we went over today, how to read plasmids and look at plasmids and things like that? Sure, Jeff, that might be possible. Um, the other plasmids probably have, because they have different promoters, the sequences are going to be slightly different of the promoters in those other plasmids. But that is a possible way to try and find out the exact ProK1 promoter. Yeah, I mean, so you probably don't need to necessarily even use CRISPR um, to make GFP cells. You can probably just use a plasmid. So one of the cool things is if we go to AddGene, right, and we say we want GFP, right? We just want the GFP gene. All right, now what we can do is we can search, we want GFP for mammalian cells, right? And let's see what else. We want human mammalian cells. Is there any other things? You can choose your selectable marker here. Um, Most common ones are probably neomycin or blastocidins. We can go neomycin. And yeah, let's see what we got here. So some of these are going to be GFP that's connected to other things, which isn't necessarily what we want. Let's say codes 1 insert. Now we just have to find a GFP. So usually when it has like a name with GFP and then something, it's like GFP linked. So a lot of times they link GFP to other genes because then it makes it easy to visualize them. So it might be a little bit of searching to do to find ones that just have GFP. One thing you might be able to do is throw in something like eGFP. Let's see what happens when we do eGFP. Oh, here we go. pcDNA3 eGFP. That should be just GFP. So theoretically, you could take this plasmid, grow it up in bacteria, purify it, and then transform it into human cells, and it should make them fluoresce green. It should put GFP into the human cells. So there is not much difference uh, between there's not much difference between the SnapGene viewer and the SnapGene full paid version. The difference majorly is that if you want to edit the sequence, so say you're trying to create a custom sequence in here and you want to just like, let's say we just want to delete these bases out here because we're trying to create a custom sequence. You can't do that without the full SnapGene version. But what you can do if you want, right, is just copy the whole sequence, paste it in Notepad, copy this sequence, find the sequence in Notepad, and then just delete it, right? So while that is, a good benefit, I think it costs like $300-400 per year for use. Um, I would say right now save your money and you know it'll be a lot easier. So Jeff, if you CRISPR stuff in the genome, it does become more permanent, but CRISPRing things into the genome is a lot more difficult. So usually when you put a plasmid in a cell, as long as you have a selection, uh, as long as you're selecting for it, it should be fine. And like I said at the beginning, even generally when you're not selecting for it, you could get, like, the plasmid will still generally stay in there, at least for one generation or a few generations. It's if you keep trying to grow generation upon generation of the organism, eventually they'll lose it. But can we use, I don't know what GES is. Is it like genius or what? Genius. I mean, you could use whatever you want. I think SnapGene is generally just the easiest. It's the most commonly used amongst scientists and people. So that's why I use SnapGene. That's why I'm teaching you how to use SnapGene. You can use anything, whatever you are used to. If you're used to it, if it works for you, use it. Use whatever works for you. I don't mind. But I like SnapGene. It's just really easy. It it's super easy to use. It allows you to import stuff directly from AddGene and NCBI, which is also really nice. So, if you just need a plasmid or something, it's really good to just get something from AddGene. Uh, but yeah. Yes, VMD is a protein visualizer. We'll probably not really go over proteins that much in this course. This course is more about DNA. So we'll focus on DNA, maybe some RNA, but mostly DNA in this course, Jeff. So we won't talk much about proteins, at least not that much. All right, any other questions? So what I would suggest is just get your handle on using SnapGene. Um, if you haven't received your package yet, it should arrive any day now. And the package should contain the GFP plasmid and the PDAWN. So what I would suggest is get familiar with those two plasmids, GFP and PDAWN. Um, just look at them, understand what you're working with, just have a general idea of what's going on. And next week, we will begin experiments. Next week, we will start talking about growing up bacteria, purifying DNA from bacteria, and all that good stuff. So we can get going with the experimentation. All right, sound good? If you have any questions uh, about SnapGene, feel free to post them in the Google Classroom. Um, you can also find me on Facebook or things like that, and send me questions there. But the best place is probably Google Classroom because we have a couple teachers, um, Esther and Jill, who are not here right now, but will hopefully be helping out with the class in the future. And let me know what you all think about this 5 o'clock time slot. If it's good for you, or something else. Otherwise, thanks for joining, and I will see you all next week.